Hello, this presentation is going to cover RABS, isolators and containment, the technical essentials. We'll be looking at isolators, aseptic material movement and the Annex 1 implications. So, what do the contents cover? Firstly, who am I? What does this presentation cover? Back to basics, design features and principles, material movement, cleaning and sterilisation, Annex 1, the key takeaways, what the FDA say about sterile containment and the financial impact. So, firstly, who am I? My name is Ewart Richardson. I've worked all my life in the pharmaceutical industry. My background and qualifications are in mechanical engineering and business management. I spent my early years at GSK working in both R&D, technical development, engineering management through and into major projects. From there I formed my own business called Isolation Solutions, which was bought by DEC in Switzerland, where I worked as Global Technical Director. I worked on and run many projects within the pharmaceutical sector, both globally and locally, including working with companies including GSK, AstraZeneca, Pfizer and many more. Also key suppliers such as EMA, Harald Hofliger, Bosch. Also minor and major project management houses including Jacobs, Project Management, Cytec and many more. Latterly, I've consulted for a number of businesses including GSK, Norbrook and the UK's own National Health Service, to name a few. I've worked with AUSTAR for many years and I'm on AUSTAR's Technical Advisory Committee. So, what does this presentation cover? This presentation is not going to cover the entire subject. To do so would take much longer and it would not do it justice. Containment technology is now a mature and accepted technology, but that's not always been the case. Many years ago when I started looking at containment technology, it was new, very little was understood about it, and the key people that were involved in it, many now have retired. However, it now touches many aspects of the pharmaceutical industry, both in production and research, including both sterile and the high potent production of products. It has now been enshrined in the new EMA Annex 1 regulations, which is in its final draft, which will be released this year in its final form. This presentation will cover the basics and the critical requirements as Annex 1 as I see it. So without further ado, let's begin. Back to basics. Why would you even isolate in the first place? 4 key reasons. Toxicity of the product. Protection of the product, either for sterile reasons or for a specific processing environment. Protection of the operator. And certainly something that is often forgotten, economic. If this is done properly, it will give you a significant payback, and we'll cover that later on in this presentation. So, if we move back to basics, for those who are unsure, and I'm certain some of you will be, what actually is the difference between a RABS and an isolator? So, a RABS, it's effectively an open system where you have a cascade of air moving down through the unit, blowing over the equipment with the air passing out at the bottom. Now, this is not a closed system, but it's what it says it is. It is a restricted access barrier. So what you are doing is restricting people's access to the equipment, but using an air curtain effectively to blow down. That air curtain has to be around about 0.45 meters per second and will be classed as laminar flow, though the new guidance does give you wider perspective on that, between 0.36 and 0.45 meters per second. It also allows you to produce your products in a facility that is 1 log down. So you would, if you were producing in grade A, you could put the RABS system in a grade B environment. If we look at an isolator or a containment system, that's different. At this point, you are closing the whole system out. And if you look at the diagram on the screen at the moment, you will notice that you can either have turbulent flow or laminar flow. Generally for sterile, laminar flow is accepted. However, you can use turbulent flow if it's going to have an effect on the product or if it's going to cause generation of particulates inside the unit, which is going to be more dangerous than the sterility itself. You will also notice within the diagram that you've got access to manipulate the processes from glove ports to half suits. You'll also have airlocks, rapid transfer ports, VHP for sterilisation. So there are many aspects to it which I'm certain you can see, and if you look at the photographs you will see some of the pictures of some of these facilities that are inside a classical isolator. A RABS or an isolator can be a complex integrated system, and you can see within the picture a sophisticated system that was built across the 3 floors, which includes isolators of various types to suit the process. It can be built around a process, so you are covering the process and having that within an isolator. You can even have flexible systems made out of flexible materials, although this does take a little bit more thought on how you construct and the implications of how you sterilize this and the risk of those materials cracking as part of your process. Around filling lines, I've done this on many occasions, completely around filling lines or to test products. Certainly this is not the only ones or the only systems that are available, but that gives you an idea of what can be used around processes. Key design features and principles. Obviously, for toxic products, isolators are perfect for handling toxic products. These are generally negative pressure isolators, typically -50 to -150 pascals. Isolators can be closed, or the word is open, but that doesn't actually mean they're open, but they're slightly open. That would ensure that any materials are kept in. For sterile products, the isolators are both aseptic for processing. There will generally be positive pressure, but I'll also put that within quotation marks because that's not always the case, because it can be positive to the process but negative to the environment that it's sitting in. They would typically be between +25 to 100 pascals, and they would be generally fitted around machinery or some of the sterile testing environments. RABS almost always are around filling lines. I would say it's an older technology, and I'll go on to explain that now, but that's a hangover from past ways that we used to produce. I personally am not convinced they're necessarily the right place to go, but they do have a place in the industry for lower cost processes. In particular with RABS, you really need to use first air principles to ensure that the airflow covers the product, and it would remain part closed around that area in the surrounding environment. Now, if we consider the key principles when you're looking at containment, and the reason I'm discussing this is because some of these flow over into sterile production. You'll be looking at elements such as the clean room classification. You're looking at A, B, C or D, and remember, for an isolator, one of the key takeaways that I'll discuss later for isolators is the benefit is that you can process in a Class A environment but operate in a Class D environment on the assumption that you've done your risk assessments correctly. Again, I'll discuss this later on. Toxic level banding, generally it's been 1 to 5, but sometimes people are using a 1 to 7 range now, as you can see within diagrams. The operator exposure limit, the sterilisation method you're going to use, obviously that becomes more challenging with RABS, but again I will discuss that later on. The ergonomics, making sure you can reach every part of the system, that is absolutely critical, and ergonomic modelling is key to any successful process. Any special processing conditions you might need to worry about. That could be anything from moisture, or it might mean a nitrogen blanket if it's explosive materials. The materials of construction, generally stainless steel, but you may have to consider other areas if you've got a highly acidic product. Process equipment integration, how they're going to integrate with the system so you can clean, you can disassemble, you can reassemble effectively. Any cleaning, whether you're using solvents or detergent or just WIP systems. Obviously reviewing of operability and ergonomics, and flame-proof and dust generation issues, which obviously may bring in ATEX within to some of this. So, understanding the applications and high potents. Understanding containment for isolators and high potent compounds. And why am I talking about this when this is generally a sterile presentation? The main reason is very simple. Many of these will apply to both. However, if you understand the differences, it will help you in understanding what you need to do for your processes. So for high potent compounds, generally, as I say, operate under negative pressure because you want to ensure that the dangerous product remain inside the working environment and any operator and the facilities are protected. Typically that will be a grade C, class 10,000 at rest. But if you're sterile, obviously you'll be at class A. There must not be any exchange of air with the surrounding environment unless you are passing through HEPA filtration or occasionally through ULPA filtration. The equipment may be equipped with nitrogen because you may want to suppress any explosion due to the nature of the product, because quite often if you have a product that will go airborne and is very, very small, you need to think about that, and sometimes nitrogen is a way of suppressing that. All materials exiting the isolator need to be cleaned and contained. So you're looking at things like rapid transfer ports, bag out ports. I will cover these technologies later when we start looking at material movements from and out of these units and into these units. Typically, they must be cleanable, and this must be reproducible in a quantifiable manner. And generally, that will be swab tested during qualification. Further things that you need to consider within these units are it's got to be having interlock doors, and again that is something that's becoming part of the Annex 1. They expect to see interlock doors on some of these systems, but that will be primarily for sterile, but certainly I would look to do the same for high potent products and high potent systems. Visual display to indicate the status of any alarms on the system. I would also expect the radii in the corner of the system to be at least 20 millimeters. The chamber fittings require a good 2 to 3 degrees of fall for good drainage, though I've seen some fall at 5 degrees, because sometimes even 3 degrees when you test it isn't necessarily enough. I would also expect a leak test to be available to be done before you start running the process. Now I would expect that to be the same on sterile units as well, and in fact it's something that is required in Annex 1. Also, I would expect to see glove pressure tests tested prior to manufacture. Again, I would expect that to be the same as Annex 1 now calls for that for sterile systems. So there's a lot of carryover from the two. Also for high potents, you wouldn't necessarily be looking at laminar flow. You'd be looking at probably turbulent flow, but making sure there are no dead spots in the isolator. But that means you'll be looking at possibly push-push filters, which obviously are much safer to change than laminar flow units. And also looking at systems that safe load products into and out of the system. So again, rapid transfer ports without compromising the surroundings. Now let's look at specifically aseptic isolators. Again, you can see where some of the wording and some of the way it operates is going to be exactly the same. So again, they must not exchange air with the surrounding areas. So you would expect to use ULPA or HEPA filtration. Typically, for a sterile isolator, you'd be looking at grade A. And if you look at the table, you can see the particular standards that you'll be looking at for grades A, B, C and D. Benefit for an isolator in this, unlike a RABS system, you can operate a grade A isolator inside a class D facility, which significantly reduces your operating costs, but that must be risk assessed. I cannot emphasize that enough, and that's certainly called for within the new Annex 1 guidance. Typically these systems will work under positive pressure, because what you're looking to do in an isolator for a sterile unit is to push any particulates or any risk of ingress away. So if you have any issues, the system remains safe. Also, they must be sterilised in a reproducible manner. Now usually this is done with VHP, and again I'll discuss this later on, and it's quite odd within the new Annex 1 regulations, VHP isn't specifically mentioned. It mentions a sporicidal agent, but it doesn't mention VHP, which is odd because nearly all isolators that I'm aware of that are sterilised generally use VHP. All materials that enter the isolator must be sterilised and enter either through a decontaminating route or a sterilisation, which may include something like a depyrogenation tunnel or via a rapid transfer port from a previously sterilised process. For sterile toxic applications, generally positive pressure with lockdown process phases with optional turbulent flow or negative pressure, which is risk assessed, should be used. And if you notice, you can see it's within Annex 1 14.19 within the regulations. Now what I'm saying there is that because it's sterile and toxic, you have a double problem. You need to keep things out while keeping things in. So it's looking at how you balance the systems to achieve that situation. It's not easy to do, and generally relies on complete control of the system and either using your airlock as a plug, which means you're pushing things in and out, or as a sink where you're pulling things in. And to do that, to make that decision, you really have to look at doing a full risk assessment on the process and getting full understanding and challenging it at the design qualification side. You'll be looking at obviously interlock door systems to assure that you do get a plug or a sink. You'll be looking at visual display indicators on the status of the alarms in the isolator, i.e. it's within the pressure range that it needs to do to operate correctly, along with your airflows inside it. We've discussed having radiused corners to make sure it remains clean, and we've discussed the 2 to 5% drainage. Again, the leak tests I would expect for each processing routine, the glove pressure testing I would expect to do, laminar flow or push-push filters. The push-push filters would really need to be tested if you're going to go for a turbulent flow system, to make sure that you've got good flow of air in the system and you're looking at test visualization. I've built several isolators doing it this way. It's challenging, but you need to do a lot of work up front again on your model of the system to make sure your airflows very much cover inside the isolator and there are no dead spots. And also we've spoken about loading systems using rapid transfer ports. All of these you can see within the pictures on the other page. Well, what about RABS? RABS, as I said before, are much more simple to deal with in this case. Certainly you would not be looking at anything other than something where it blows air over the system. You'll see in the pictures that you've got different types of RABS system, but unlike an isolator which is closed, RABS are not. You can say that when you get to sort of a closed RABS system where you have filters at the sides and the top, you're getting close to an isolator, but you're still not a fully contained system. And the reason I talk about that is because from my experience, the biggest issue you get inside a RABS system, if you're going to state that it's closed, is air penetration and particulate penetration through the bed of the machine that it's covering. That's very difficult to achieve and needs a lot of work with the machinery supplier to achieve that. And it's something that you really need to consider. Certainly, again, you're back into this risk assessment situation that as part of your company contamination control strategy you would have to do, and that's certainly stated now in Annex 1. And the downside of a RABS is that really, even if you've got a grade A RABS, you really should only be producing in a grade B environment, which has a lot more cost implications in terms of when you run this system, and you'll see this later on in this presentation. Material movement. So we've now got an isolator. How do you bring things in and out? Now, that's always been one of the difficult challenges, whether your system is sterile or toxic. How do you introduce items into a closed system, and how do you remove them safely out of the system without compromising? Many systems have been developed to allow this to happen, and many more are still being developed currently. And there's a few new ones on the market, again, which I'll talk about later on, that may solve some of your problems. So your products are going to come in a variety of forms. So there are solutions for transferring of powders, there are solutions for the transfer of liquids, there are solutions for the transfer of components, the transfer of products finished products leaving the system, and the transfer between process stages, and the manipulation of equipment during production without compromise to the external environment. So what do these look like? If I look at something, this certainly doesn't cover every solution, but it will give you an idea of the solutions that are out there. You can look at things like the powder transfer system that was developed by DEC. Now, there are many companies now starting to provide this because it's, I believe it's now at a patent, though the DEC system is probably one of the best. What it's really doing, it's a powder pump. It's moving product from one process through the PTS system and down into perhaps a vessel, perhaps a feeder. It could be into anything. But it allows you to completely move it without any contact with the outside environment at all. Obviously, there's the traditional rapid transfer port system, which have been around for many, many years, originally developed by La Calane, but again, there are now many, many systems out there that have taken that technology a little bit further and built maybe slightly better systems, more robust systems out there. And that uses a simple, effectively door within a door system where the two come together, and as they're locked together, the whole door opens together. There's also split butterfly valves now for transferring powder between systems. They sort of cheat the sterile system by docking together and then allow a wash or a UV sterilization between the two faces before they finally clamp and close. So doing what the systems that are used for sterile toxic, but perhaps are cheating it. But they are available. Obviously, we have the simple airlocks where you have a pressurized airlock, which we've discussed, to plug in and out. But again, I think it will depend very much on what you're trying to achieve and the level you are on. And I keep coming back to this, doing your risk assessments properly and making it part of your contamination control system. There are new systems coming on, and you can see that on the slide. There's a company called EasyDoc that do a system for transferring powder between bags in a safe, secure way, and there are others. So this is an ever-evolving technology that's now being used, that's now increasing its options for customers that are out there. Transfer of liquids. There's obviously the SART system, which is developed by Sartorius, which effectively plugs 2 points together, which you can see in the diagram. That's a very successful way of transferring between vessels into filling lines. Obviously quite challenging quite often. You can even use the liquid PTS, which pumps liquid between 2 processes. Very good, very simple, but maybe not as delicate and as measured as some could be. And I would say there is the old method of tri-clamp connections under laminar flow to allow liquid to pass down into your filling line to fill into your ampoules or vials. Transfer of components in and out. Now, again, this is something that's always been challenging. So a lot of the technologies are using some of those to transfer powders and components in and out. So if you look, you can see rapid transfer port bags. That's exactly what they are. So what you would do with that, you would possibly pre-fill your bags, quite often send them off for gamma irradiation with possibly your stoppers inside them. They would be irradiated and sterilised, and then following that they would be docked onto the isolator, and then in exactly the same way as a rapid transfer port works, they would then pour into the isolator into the vibratory bowl feeders that start to go on the filling line. Now, I've seen them used both on RABS and on isolated filling lines. But that is one very, very good technology, single use, very, very effective. For compounding isolators and intermediates, and generally you've got the rapid transfer ports that do that job. And again, we've discussed those. They're very common technology, and I'll be surprised that anybody here that's listening to this hasn't used those before. And obviously we've spoken about airlocks and sterile access systems, which again are used depending on what process you're looking at. Again, all these, and you'll hear me reiterate this repeatedly, would have to be risk assessed on which is the most appropriate. And I would expect any business to look at this at the very early stages to start understanding what is the right solution for your business, your process, and where you are at this particular time in your cycle development. Transfer between processes. Obviously, you've got rapid transfer ports, which have been around for many, many years for moving components in and out and testing materials in and out of an isolator. There are new technologies such as the EasyDoc system, which allows the transfer of powders through into isolators and sterilising the faces of the connection device. If you're looking to move vials into a system, then obviously there are depyrogenation tunnels and washers, obviously pre-wash the vial and then go through the depyrogenation tunnel. But be aware, with a depyrogenation tunnel, you're likely to get a log 3 kill result rather than a log 6, which is typically expected for sterile systems. That's recognised, but some companies may tell you that you'll achieve log 6. I'm dubious about that. I think it's something you'll need to test as part of your validation program and pick up with the supplier during the design qualification period on the project. Obviously, we've got airlocks, and there are some odd systems such as the batch mixer from DEC, which will allow powders to be blended and then moved into a secondary process. And there are other unique ones out there. Transfer of components continued. We've mentioned depyrogenation tunnels and washers. Also, there is the old technology of bag out ports. I've used these occasionally. They're very simple, very effective, but again, you have to be very careful how you use them. And I would probably look, if I'm taking materials out through or intermediate canisters out through a bag out port, to possibly have an intermediate chamber before that to ensure that you've not compromised any sterility. But they work very, very effectively. Transfer of finished products out. Obviously, as vials leave a RABS, you would generally have an airlock, as you can probably see in the pictures on the presentation. That would allow airflow over the key areas to come out, but at that point, either the ampoules are sealed and closed, or the vials are capped and stoppered, so they're quite secure at that point. There are many other ways of doing that, as we've spoken before, from pressurized tunnels. The same principle is used for blow fill units. But again, it's looking at what you're trying to achieve. Now, generally, it's actually quite easy to get finished products out because at that point, the processes are closed, but you'd look to have a staged removal of the finished products, so there's no risk of compromise of any airflows or any, should we say, compromised air moving backwards through the containment system. So care has to be taken on that. That may be done by doors, or it may be done by pressurized systems within your process. Manipulation into these units. Well, obviously, the general principle is to use glove ports. They're used everywhere. The things to consider with glove ports are both the chemical resistivity and the material resistivity. Now, that comes down to your operators and how aggressive they are with these systems. You will puncture these gloves at some point, so you do need to have safe change possibilities, which obviously need to be risk assessed, whether you lose the batch if you do have a compromise on your gloves or not, and that's something you need to factor into what you're doing. Equally, I have used half suits on these processes. Now, these come in very well when you have a complex process where you cannot reach certain applications, because glove ports work very well, but you do have limited reach. In general, 500 millimeters is your limited reach within a glove port. A half suit gives you significantly more, probably up to a meter around a bubble around the operator. They're not used very regularly. It is something that I wouldn't recommend to do unless you had to, but it is possible to use these systems, and they do work very well, provided you've looked at the materials of construction, you've looked at cleaning, you've looked at safe change, and all the other aspects that you would look for in a glove port. That would include pressure testing them on a regular occasion to ensure they've not been penetrated, which would be the same case in the glove port systems, where you'd be looking to pressure test your gloves. Generally, after I would think either every batch or certainly every campaign. New technologies that are coming along, and I've seen this now recently, we're looking at robotics inside an isolator. Now, obviously, the benefit of having robotics inside an isolator are that you are going to contain the process even more, and there is absolutely no human intervention. The only challenge would be cleaning these units and how well they'll stand up to the sterilisation process as they operate, and can you maintain the process? Is the software good enough to be validated and part of your risk assessment, part of your validation process, and part of your validation of the unit around GAMP, because that may be a possible struggle depending on who you're using, though it's certainly a technology that's going to become increasingly and more widely spread, and certainly would guarantee any of your aseptic processing in a much better way, and would remove people from that process, which, as we all know, are the biggest risk. Cleaning and sterilisation. Obviously, we've discussed this within the isolators before. Oddly enough, and for some reason, Annex 1 does not mention VHP specifically. Really, really odd. This is the most common way of sterilising an isolator. So once your process is finished, you've gone through your clean in place cycle, you've taken all your swabs. To sterilise it, normally you introduce a vapour into the isolator to sterilise it. Some of the key things you need to consider when you are doing this phase is, obviously, there will be a degree of cycle development to understand the best cycle, but the biggest issue that I've seen is how processes and how equipment is laid out within the isolator. Bear in mind what it actually says: vaporized hydrogen peroxide. So we're looking at a vapour. That vapour has to get to every surface within the isolator. That will include any components within the isolator it has to get to. So if you've got a component that's lying on the surface, then you won't get a decent flow of hydrogen peroxide under that unit. So you need to have hangers inside, you need to have it separated, split, so you get this around. Generally, the units can be integrated into an isolator itself as part of the control system, but most of the time you'll find it will be a simple unit like the one you're seeing in the picture, and it will then take control of the isolator, and what it will do, it will dehumidify the isolator, because obviously you don't want any humidity inside. It will then condition the isolator, and by that it means bringing it up to temperature so you're getting a good vapor phase. Then it will hold the vapor phase and the heated temperature inside the isolator for a period of time, and that will be developed during your cycle development to ensure that you get a maximum kill. And then once that's finished, it will go through an aeration phase, which is removing all the hydrogen peroxide out of the system, and would then use a catalytic converter to bring it back to water and oxygen to leave the unit. And then you should have a sterilised unit. You can use them in flexible isolators. My only concern is over a period of time that quite often causes a plasticizer to be removed, and you may get cracking on the flexible systems. So personally, I would say if you're going to use them only inside a solid isolator that's probably obviously a stainless steel construction. RABS, the cleaning process is obviously slightly different because the units are open, and because they're open you certainly cannot use vaporised hydrogen peroxide or any significantly vaporised sporicidal agent of any sort for all the obvious reasons. So as they're open system, decontamination becomes much more challenging to be repeatable and validated, and generally you'll find there'll be liquid wipes on the inside, which again is why RABS in my viewpoint are a little bit more challenging. The other introduction that you'll have to bring into a RABS, generally you'll need a parts washer, because any parts on a line cannot be washed in place. You'll need to remove them. I mean, this certainly could be the case with an isolated system, but absolutely definitely with a RABS system where any spare part or any parts, change parts, will need to be removed, taken out and cleaned and then brought back in. And that obviously then presents another challenge with how do you bring these parts in and out of the isolator without compromising sterility, which is something that you need to think about as part of your decision when you actually decide, do I go for an isolator, which technically will be more expensive, or do I go for a lower cost RABS, and what are the effects on my process? Obviously Annex 1 is about to be released. It's in its final draft. It will be released later this year, and that will become enshrined in the regulations for the production of sterile products. I'm going to go through the key takeaways that I feel are important, but my viewpoint might be slightly different to other people because each person has their own specialities and areas that they feel are important to them. Though, having said that, every item within this is very, very important. It's a huge subject. Without doubt, I would not even intend to cover this in this presentation. It's far too big, far too complex, but I'll look at the things that relate to containment that I think should be looked at if you are involved in this technology. So what does it say? What does it mean for containment? The first thing it means is you must have a good contamination and control strategy. Now that sounds like something very simple. Trust me, it is enormous now, and I'll go on to discuss this later on. There are a huge number of elements that need to be covered within this to make sure you've got a robust process, robust contamination control strategy. I've been in a few plants recently where they hadn't, and they were very much picked up by inspection agents for not having a good process in place and something that was registered on their system. You must obviously, your environmental monitoring is now part of that process but separate too. So I'd also add, and it's not part of the Annex 1 documentation, but my view is it should also cover anything that relates to high potents, because you will get picked up if you're not looking at your high potents for all the obvious reasons, and how you control them from goods in right through to your finished pack form. All your processes now, as I've stated before, must be risk assessed, and I'll go on to discuss what that means if you're not aware of it. You cannot just rely on guidance. So in fact, some of the guidance actually says you have to develop your own standards to make sure they're right and have been risk assessed. This has implications for the design and the operation of both existing facilities and or new facilities that are being considered to be constructed currently. Containment guidance and RABS guidance is now part of the regulations. That to me I think is fantastic. They are now enshrined in this document, and there's certain things you have to do. And as we said right at the start, a grade A RABS process can now be installed in a grade B environment. A grade A isolator can be installed in a grade C or even a grade D environment, but they all have to be subject to risk assessments, which will form part of the business quality risk management and can be audited. So bear in mind, this is now going to be audited when previously it may not have been. Now, what do some of these techniques look like? I'm certain most of you who are listening to this or watching this presentation would have seen some of these things or been involved with them. Certainly failure mode effects analysis. Yeah, we know they're dull as dishwater to do, but they have to do, and they're very, very thorough. That's the sort of thing you'd be expected to do. You may be looking at fault tree analysis. You may be looking at hazard analysis of critical control points, maybe having a hazard analysis plan, or even on when you're looking at the more issues with safety and process, you may look at having a basis of safety document or even doing a HAZOP. So there are many ways of doing this. There are many techniques of doing it, but these processes are now going to be audited by inspectors and are specifically stated in Annex 1. So it's something you're going to have to do now. Now, as I said, the contamination control strategy should at least reference the following items. Now, you can see how big this is. So you'll be looking at design of both plant processes, including all associated documentation, the premises and equipment. So you're not just looking at the equipment, you're looking at your premises, how you move between inside the premises, how you move your materials, what it's manufactured from, where you clean them start and finish, all of that. Your personnel, how they're going to behave, even how they're going to behave inside a grade A environment, hygiene, changing procedures, entry into units. So it's going to be much wider, and you can see all these within the documentation. Utilities now, your utilities will form part of this. So anything that touches the process in any way, shape or form, and this could be WFI, this could be your HVAC systems. So any of those will need to be looked at and would now form part of Annex 1. Control of your raw materials, including process controls, that needs to be now part of this. Product containers and closures, vendor approval. Now, that means that you're going to have to treat the vendors as part of your manufacturing process, because they provide key components, and that might be, say, stoppers, it might be caps, it might be single-use systems. You're going to have to audit them as if they were part of your business, because they are part of your supply chain. So significantly more work needs to go out to do that. I've seen some companies where they just send out a postal vendor approval system. I personally think that won't be good enough. You'll physically have to go and see these companies, and that may be that you need to travel around the world to do that. But unfortunately, that's what people are expected now, and personally, I think quite rightly too. Management of any outsourced activities. So that might be any contract services you're using. So that could be validation, that could be maintenance. That will be the same way as you would treat your key components, because these companies that are doing this for you obviously have a profound effect on your product quality and your management of your processes. So again, they all need to be audited and checked in the same manner. Process risk management, process validation. Certainly Annex 1 refers to Annex 15 and Annex 11, which relate to validation, and there are certain expectations that you have to validate your processes too. That's all recorded done. Obviously, validation of your sterilisation processes, as one would expect, whether that's steam, VHP, depyrogenation, whatever. You're going to need to do that now and make, again, ensure it's validated. I'm certain anybody looking at this would have done this anyway, but there'll be much more emphasis on that. Preventative maintenance, maintaining equipment, utilities premises, and planned and unplanned maintenance. You'll need to put that as part of your contamination control strategy. Cleaning and disinfection, obviously. Any monitoring systems, including the assessment of the feasibility and introduction of scientifically sound and alternative methods that have been optimised, the detection of environmental contamination, you're going to need to look at that. So what that's really saying is any way you do this, where you put your settling plates, how you set your facility up, where you're doing the testing, what parts you test, the airflows you're looking at, that needs to be recorded and be very, very robust. Also, there'll be an expectation to do trend analysis for your detailed investigation. So if you have a problem, you're actually looking at trends to see if that problem's getting worse over periods of time. Obviously, building management systems are great for this. SCADA systems are great for this. Equally so, you could do it, should we say, semi-manually. Again, there'll be expectation that you'll be doing root cause determination, RCAs, on this. And also, when you do a problem, that a CAPA will be raised and you'll be looking at comprehensive investigational tools to determine what you've done wrong and how you found that problem and what you need to do to put that right. And this will all be part of your continuous improvement process. So this isn't an inclusive list, but the salient points. So you can see this is going to be a very, very big document, and it's going to cover basically all your facilities and how you operate your business. And it's something to be aware of. And I'm not sure that every business globally necessarily does that to the way that possibly the larger companies may do now. If I look at following guidance, so this is going to be a big list, but within that 59-page document, there are also expectations around pharmaceutical quality systems, PQS side. There's expectations, and I've put the sections and the pages within the document so people can find it easily enough. There are expectations around premises, expectations around barrier technologies, which we've obviously discussed in this presentation. There will be clean room and clean air qualification on how to do that, and some of that's new. And I think I said earlier that within that there are certain standards that you may want to achieve around clean rooms, where the actual guidance doesn't tell you what they should be. You have to derive them from your own risk assessments. There are specific sections on disinfection, equipment, utilities, water systems, steam systems, gas and vacuum systems, heating systems and cooling systems, personnel, production and specific technologies, environmental monitoring and quality control. So you can see this is a huge document covering virtually every aspect of sterile production. So be aware of that. I've only focused on a few items that relate generally to containment. So what does the FDA say about it? Now this is a statement that came from Rick Friedman, I would say probably 10 years ago, and he's mentioned it before. Rick is their Deputy Director of Quality Manufacturing. And if you read what he says, effectively, he's really saying that the FDA will look much more closely at businesses that do not use either RABS or containment systems. The reason for that is they have found far more non-compliant facilities that do not use these systems. So effectively they're saying, if you're not using these systems, they don't believe you're in control. Now that will be the same logicality now within the EMA and the UK's MRHA and many inspection authorities around the world, which is why you're now seeing the systems or the containment units being brought into the Annex 1 regulations. Rick is very well respected in the industry, and he's spoken about this many, many times. So take that on board. If you are not within a containment system, if you're not within a RABS and you're producing sterile products, I think you're being foolish and you're running the risk of having your business seriously inspected and possibly shut down, or having certainly a significant 483. So my personal view would be, I would look at all my processes now, particularly as it's called for within the regulations, to actually either be in containment or RABS of one form or another. Now, the thing that comes out of this really, the argument I've always had thrown at me is, well, we can't afford to do it. We can't afford to change things. So let's have a quick look, and this is one of the last slides, at the financial impact. I was very, very privileged to be at a conference many, many years ago, well, not so many years ago now, with Boehringer Ingelheim that were doing a presentation. And I've taken this one slide from that presentation to make the point. They were very fortunate that they were producing two sterile filling lines next to each other. One contained and one not contained. And, well, so say one isolated and one in RABS. Obviously a RABS is not as well contained as an isolator. You'll notice the figures for the capital depreciation, i.e. the cost of that unit over the year, are very close. So for a RABS you obviously build a clean room and all the aggravation that goes with that. For an isolator you can reduce the standard significantly. But if you look at the depreciation, roughly the same to build these units over the year. However, as you run through the presentation, if you look on the right hand side, you'll see the cost for monitoring over the year. You'll notice that the cost for the RABS is nearly 2/3 of the cost to actually build it. So if you go through into the second year, you spent more on monitoring and testing with a RABS than you would have in an isolator. So it becomes significantly cheaper to run an isolator, and you'll get significantly less arguments with inspectors if you're inside a well-designed, carefully operated, well-presented isolator system. So the argument that companies have saying, oh, we can't afford to do it, I'd actually throw the argument back the other way. You can't afford not to do it. I've been involved in this as a customer to the industry, certainly back in my early GSK days and since then, but I've also been a supplier to the industry. I don't know anybody who's put isolator and containment technology in that hasn't got a significant cost reduction in their processes. So as a final point to this presentation, you've now had the technical essentials around isolators, RABS, containment systems. You've had the new regulatory advice coming out, which is going to tell you you're going to have to do it whether you like it or not. And you've now had the cost argument that clearly shows that doing it inside an isolator is significantly the cheapest way of producing your products. So I can't think of any reason why you would not, and I think you'd be foolish not to do so. If you have any questions, please ask me after this presentation. Thank you for listening. Hopefully speak to you again on other subjects.